Good day, dear students. Current lecture will start new part of microbiology. It is known as virology, and now we will talk about viral infections and viral diseases. In first lecture, we will discuss one of the most famous group of viruses. It is respiratory pathogens. And before we'll start any discussion of precise virus, we need to refresh some knowledge on common virology. What is virus? And of course, you should remember, you should understand that it's absolutely unique type of microorganism which have no cell organization. It's absolutely differs from bacteria, and common knowledge about bacteria are not applied to viruses. So it is again non-cellular type of microorganism. It means that virus have no cell. It have no cell organelles, no cell structure. It have no properties of the cell, and actually it can replicate only in another host cell in a living cell, because virus is obligate parasite. It's not able to multiply and to perform any activity itself. It needs a host to use it. And if you remember that classification, that virus actually exists in two particles in two forms. It is extracellular and intracellular form. So while virus, we call it virus, it is commonly in medical world and actually in common community. But actually that microorganism which is identified outside the cell, which is transmitted from person to person or from one host to another host, it is virion. It is complete. Particle contain all organelles or all compounds of that precise virus, and it cannot replicate. It can only infect a host cell. And while host cell is infected, and when host cell is infected and virus is inside, it is actually virus. It cannot infect other cells because it's incomplete. It will lose some organelles, some compounds of that particle, but it can replicate. It can use host cell and change the host cell for self-replication. And actually, that form is called virus. But again, in common language, we always name that organism as a virus. And of course, if virus have no cell organization, it cannot be classified into gram-negative or gram-positive. It cannot have some other common properties of bacteria. And actually, it's not possible to see a virus in regular microscope. And shape of virus, size of virus is not important in routine diagnosis. The main property which is important and which is very unique for virus is type of genetic material which is present in virion. And if you remember again, virus cannot have both DNA and RNA. It is too small and too primitive to have both types of nucleic acid. Again, it is an exception, but in your program and classic description, virus is classified into DNA containing and RNA containing. It is always one type of genetic material, and it is important in life cycle of virus that can have different types of multiplication, different places of multiplication and host cell, and they can establish very different types of infections. So virus have kind of primitive structure. Again, the main genome of virus is presented by only one type of nucleic acid. It can be single-stranded, double-stranded, complete, or segmented, but it is always some nucleic acid which is present in the center of the virus in inactive state. That acid is surrounded by protein coverage, which is known as a capsid. Capsids have unique structure in every species of the virus, and viruses are classified as well into family, genus, species as bacteria. That classification is common for any type of living organism. But capsid, it is a protection for genetic material. The main option, the main task is to protect genome from environmental factors, physical, chemicals, and other. And structure of capsid, shape of capsid, and composition of capsid is unique for every viral species. And actually, if you will use electron microscope, it is possible to see virus, of course, and recognition of viruses possible due to that unique presentation. And different viruses, can be identified even by common appearance in microscope. If we say that bacteria all are the same, we have three major groups: spherical shaped, rod shaped, and spiral shaped bacteria. And every group, it's not really possible to identify bacteria by general appearance, because all rods, they are kind of the same. All cocci, they are the same. And identification by a microscopic view is not possible. In the case of viruses, it is possible, because actually different species of virus have very unique appearance in microscope, and partially it is due to that capsid. So capsid, as you can see, is made of identical subunits. We call it capsomer, and number of capsomer and arrangement of capsomers is unique for every species of the virus, and it is stable. Any virus of the same species will have the same number of capsomer, the same arrangement. And every capsomer actually is made of identical number of identical proteins. It can be 1 to 4. In some viruses, one protein is present. In some viruses, up to 4 proteins can be present. But again, every capsomer consists of the same proteins with the same location. Proteins are acting as antigens, and they can be used in diagnosis of viral diseases, in identification. And during immune response, they acting as unique antigens, and antibodies can be produced in different proteins for every virus. So viruses, again, in the contrast with bacteria, cannot cause infection of any location. Viruses have unique structures. They are called as peplomers or glycoprotein spikes. It is a superficial glycoprotein which is acting as a specific receptor. The main task of peplomer is recognition of suitable host cell, and usually viruses have 1 or 2 different peplomers, and actually viruses can recognize and infect only 1 or 2 types of host tissues. And in that manner, viruses are classified into respiratory pathogens, into neural pathogens, intestinal pathogens, and one virus can cause only one infection. Influenza virus can affect only respiratory epithelium. It's not possible to have some hepatitis or osteomyelitis by influenza virus, because it don't have any receptors to recognize the tissue. But it can cause respiratory infections. We have hepatitis virus, which can affect only liver, because receptor of that virus are able to recognize only hepatocytes. And it makes viruses very limited to one system or few systems. And actually, diagnosis of viral infections at that point is a little easier, because you don't need to choose between any virus. If you see that respiratory system is affected, you always will have only few options to choose, because very rare viruses can cause respiratory infections. And second property of that glycoprotein spike, sometimes they are limited to one animal tissue, and in that manner, virus cannot affect human, animal. We have again classification of viruses according to host. We have human viruses, we have animal viruses, we have plant viruses. Again, we have exceptions, and sometimes viruses have receptors which are common for animals and humans. And the same influenza virus can affect wide range of animals and human as well. But very often viruses are limited to only one animal species or one plant species, and they cannot infect any type of the alive organism they want to. So peplomeres are usually presented by hemagglutinin and neuraminidase, and those enzymes are combined in two groups, hemagglutinins and neuraminidases, only because of one common property. They can cause unique action. But by structure, those enzymes are very different, and even within one species of virus, hemagglutinin can have very different structure, and they will induce different immune response. And usually that different in arrangement and structure of spikes is the key to differentiate viruses into different serotypes, because of course different type of antigen will induce production of different antibodies. So again, peplomecycline is antigens and very often used in diagnosis or in prophylaxis of diseases. So actually viruses are absolutely inert biochemically, and they don't have any other organelles. They don't have ribosomes, they don't have mitochondria or some other organelles, and they don't have any biochemical or metabolic activity. The only enzyme which can present in virus is enzyme which is required for multiplication of genetic material. And usually those enzymes are presented by RNases or DNases. And that enzyme can be packed in the structure of the virus, or they can be only coded by genome and cannot be present in virus. So enzyme is not obligate part of the virus. It can present in some species of viruses. In some viruses it can absent. And the last thing, the last structural compound of virus, which is again optional, it is an envelope. Envelope is usually done of lipids and originate from the host cell. When virus live in the host cell, by budding, some part of the host cell wall will stick to virus and will cover it. And envelope originate from host cell that have very difficult and complex structure. And again, due to that property, viruses are classified into naked if envelope is not present, and envelope. And again, if some viral species is enveloped, then any virus of the same species will be enveloped. That option is stable within one viral species, but it is optional in different viral species. So if you will think about it, if you will try to make some conclusions, you will understand that we have several important peculiarities of viral infections, which makes diagnosis of infection, prevention, and therapy very specific and unique and very different than bacterial one. So first of all, viruses are too small, they're extremely small, and usual light microscopy have not significant resolution and not significant possibilities. We cannot see virus in routine light microscope. And at that point, of course, we cannot study tinctorial properties. If you suspect that you have viral infections, it's not a point to take a sample and send to a laboratory to perform some staining technique or some visualization. We have a few exceptions when virus can cause cytopathic effect or cytopathic action. When host cell infected with the virus will have unique deformation, unique inclusions, or some properties that can be seen in light microscope. But again, it is an exception. In common way, we cannot study tinctorial properties. Virus always depend on the host cell. And if you will be asked, and sometimes teachers trying to catch you in some points, and they can ask, which culture medium will you use to cultivate virus? And if teacher asks that, it is an attempt to see, do you understand what is virus or not? Of course, viruses cannot grow on agar. They can grow on cell-containing culture medium, or we call it just cell lines. But in agar, they cannot be studied. And actually, cultivation usually is possible only in animals, in chick embryo, or in cell-containing culture medium. They're expensive. They're difficult. They require some unique equipment, which is again expensive. Laboratory doctors should have specific experience of viral cultivation. And many number of laboratories, routine laboratories, they don't have permission for viral cultivation. And cultural properties, of course, cannot be studied. You cannot have a colony of virus on agar. They always grow within host cells, and they can be recognized only within a unique host. Yes, they can form sometimes, some viruses can form unique inclusions or unique deformations of the cell. But cultural properties of viruses cannot be studied. And again, viruses are absolutely inactive. By the structure, they don't have even cytoplasm, they don't have water or electrolytes. They cannot perform any biochemical activity. They don't have metabolic activity, organelles, or other enzymes. And actually, the only thing virus can do, it can infect host cell and use activity of host cell and all organelles, all systems of the host cell to perform self-replication. And of course, on that point, biochemical properties cannot be studied as well. Don't say that virus is some MR positive, VR positive, catalyze positive. It's not possible. And study of biochemical properties as well is impossible. And in that way, classic microbiology is not applied to viral infections. And of course, diagnostic options are very limited. We cannot study activity of the virus. We cannot study appearance of the viruses. And that primitive structure makes viruses a kind of primitive. They don't produce any toxins. They don't have optional organelles. Even bacteria, we can identify some flagella with unique appearance or location, capsules, spores, and bacteria can be classified according to even presence or absence of some organelles. In viruses, no. Almost any virus have the same composition, capsid, genetic material, spikes, and that's all. They all are the same by that property. And again, usually for identification of viruses, we have only molecular methods such as PCR, and that's all. Or in some cases, we can have immunofluorescence as the option. But that's all. And ELISA and some other tests, yes, they're present, but usually therapeutics diagnostic options are very limited. One or few tests and nothing more. So even within one viral species, again, we can have significant diversity of compounds. And all viruses usually have hemagglutinin and neuraminidase, but structure is absolutely different. All viruses have capsid, but proteins are absolutely different. And it means that if in bacteria we have antibiotics with wide range of activity, any bacteria have cell wall, and structure of cell wall is identical, almost identical in any bacteria. Give antibiotic, it will destroy cell wall, bacteria is dead. In the case of viruses, we don't have such an option because proteins are absolutely different. And usually, if some company will design a drug, the drug is active only against one species of the virus. Even when we will talk about herpes viruses, the same family, the same structure, and the same composition of the virus. But drug which is active against one type of virus is absolutely inactive or have very weak activity against another type of the virus within the same even some family. And it makes treatment of diseases absolutely expensive and difficult because companies will need to design one drug which can be used only for one disease. If antibiotic can be used for a wide range of diseases, then here it's not working. And the main idea, viruses have absolutely another structure, and antibiotics have absolutely no effect against viruses. And if you will think when you will be asked about treatment or when you will need to prescribe treatment, it's not the point to give antibiotics against viral diseases because antibiotics can destroy cell wall, but viruses don't have a cell wall. Antibiotics can inhibit ribosomes or block ribosomes, but viruses don't have ribosomes. They can affect synthesis of DNA or RNA, but virus is not performing synthesis. Only human cells can perform or animal cells, but they have absolutely another enzymes and absolutely another structure of ribosomes as bacteria. And viral diseases should not be treated, it should not be prevented by use of antibiotics. We have absolutely unique group of drugs which called antivirals. And again, number of antivirals is very limited. Usually only some essential and life-threatening diseases have some therapeutic options. We have lots of drugs against HIV infection. We have multiple choices for hepatitis B infection. We do have few drugs against influenza virus and herpes virus. But actually, that's all. Main number of diseases have absolutely no options of drug therapy. And the only thing we have is symptomatic treatment. If subject have fever, give antipyretics. If subject have some muscle pains, give painkillers and compensation of lost fluids. But that's all. And a lot of viral diseases, if you think, they have very mild duration and mainly immunity of healthy human will do the whole job itself. And self-convalescence is pretty fine thing in main number of viral diseases. And of course, pharmaceutical companies are not motivated to have no points to design a drug because no one will buy it. Design, development, and research of new drug costs millions of dollars. And of course, company need to have some profit from the drug. But if disease can be self-convalescence and can be treated by immunity, who will spend money for those drugs? And it's another reason why main number of viral diseases have only symptomatic treatment and have absolutely no therapeutic options. So if you will remember those issues, you will understand how to manage people and patients with viral diseases, how to diagnose and how to treat diseases, and common rules of infections in bacteriology are not applied for virology. It's absolutely unique type of microorganism with absolutely unique approaches of management of those subjects.